Welcome, everyone, to today's explainer, or as we like to say, bienvenidos a esta exposición detallada. We are diving deep into the anatomy of a seriously fascinating molecule today, GHK-Cu. Now, we're going to totally skip past all the flashy skincare marketing hype and just get straight into the hard, fascinating biology. Let's get right into it. So if you've ever found yourself browsing the skincare aisle, you've probably seen this stuff pushed as some kind of miracle anti-wrinkle peptide. But you know what? The real scientific story is actually way bigger than just face cream. Take a look at this number, 31.2%. Where's that coming from? Well, researchers ran this incredible in silico analysis using the connectivity map database. They were basically hunting for a single molecule that could flip a diseased genetic state back to a healthy one. And out of over 1,300 compounds tested, this one specific molecule took the crown. Why? Because it single-handedly shifted the expression of roughly a third of all human genes. That's about 4,000 genes. It's just this massive, sweeping biological impact that makes you stop and go, wait, what kind of master switch is this? But here's where we hit a really wild paradox. This molecule, the one that managed to flip a third of our genome, is just the GHK peptide, which is literally just 3 letters, 3 amino acids, glycine, histidine, and lysine. That's the whole shebang. Physically speaking, this tiny little chain is just way too small to be acting as some complex, intricate master switch for the human genome. It simply doesn't have the structural complexity you'd expect from a molecule with that kind of crazy reach. So, how exactly can something so minuscule make so much noise inside our cells? Well, to figure that out, we actually have to look at where it came from. The twist here is that GHK wasn't cooked up in some modern cosmetics lab. Not at all. It was actually discovered way back in a 1973 aging experiment. Scientists noticed that something floating around in young human blood plasma was somehow causing old liver tissue to wake up and start synthesizing proteins again, just like it was young. It turns out your body natively manufactures this stuff. But there is a catch, a pretty big one. When you're 20, your blood plasma levels of GHK sit around 200 nanograms per milliliter. But by the time you hit 60, those levels plummet by about 60%. And that steep drop remarkably mirrors the slowdown of our own natural regenerative capabilities over our lifetime. And this actually brilliantly illustrates why our bodies respond to it so intensely. The GHK sequence is quite literally written right into your body's own building material. It's natively embedded in the propeptide of the human alpha-2 type 1 collagen chain and also in a crucial repair protein called SPARC. Think of it kind of like a fire alarm that's been built directly into the steel rafters of a building. That alarm doesn't go off until the building actually takes damage and the protein breaks apart. So when tissue is injured, the GHK fragment breaks free. It acts as a built-in distress signal telling all the surrounding cells, hey, damage has occurred, it's time to start repairs right now. It's not some foreign drug you're introducing, it's literally your own tissue talking to itself. Okay, let's get into the actual mechanics of this, because the GHK peptide all by itself, that isn't the magic. The real magic happens because this tiny chain of 3 amino acids acts as this incredibly elegant biological courier for copper. Now, on its own, loose copper roaming around in the body is really dangerous. It acts as a pro-oxidant, throwing off these highly reactive chemical sparks that can severely damage your cells. But, and this is a huge but, when GHK captures that copper ion, forming the GHK-Cu complex, it completely silences it. It shields the body from the toxicity of free copper while guaranteeing a totally safe, secure delivery directly to where it's needed most. It really all comes down to this protective transport. Now, for that transport to be totally secure, the chemical grip has to be incredibly tight. And believe me, it is. It measures at a massive 16.44. That's its chemical binding affinity, or a log K. To put that into perspective for you, GHK holds onto the copper ion with almost the exact same strength as blood albumin, which is the body's main heavy-duty copper transporter. Because of this incredibly high affinity, the copper doesn't accidentally slip out along the way. It guarantees a completely redox silent transit, meaning absolutely zero harmful reactions happen until it safely reaches its final destination. So the golden question, where exactly is this copper courier going? Well, it essentially hands off its precious cargo to act as the essential cofactor for 3 absolutely vital repair and maintenance enzymes. First up, it delivers copper to lysyl-oxidase, which uses it to cross-link and strengthen brand new collagen and elastin fibers. Second, it fuels superoxidase dismutase. This is a super powerful antioxidant enzyme that desperately needs copper to put out the fire of oxidative stress. And third, it targets cytochrome C oxidase, which is fundamentally crucial for cellular respiration and energy production. So a single little messenger is providing the exact tools needed to rebuild, protect, and energize your tissue. This whole biological process is just a beautifully coordinated chain reaction. It starts with tissue damage, which breaks down the collagen fibers. That breakdown instantly releases our built-in alarm sequence, the GHK. GHK then swoops in, grabs the loose copper, and safely transports it to those critical repair enzymes we just talked about. But here is the really fascinating part. Delivering this copper cofactor simultaneously acts as a signal to ramp up growth factors, specifically things like VEGF and FGF2. These are the growth factors responsible for angiogenesis, which is a fancy way of saying they order the rapid construction of a brand new blood supply to feed the new tissue that's repairing. It is just a breathtakingly efficient system, right? But hold on just a second. This is the part where we really need to apply some strict scientific rigor and stay completely grounded. Those striking matrix rebuilding effects you were just talking about, like doubling collagen synthesis and clearing out old tissue matrix, those were observed in vitro, meaning in cell cultures and in animal models. We need to be crystal clear here. This profound tissue rebuilding behavior has been mapped under strictly controlled laboratory conditions. We're not talking about massive human clinical trials here. Keeping that distinction in the back of your mind is absolutely vital for understanding what the real science actually is. That being said, what those preclinical models do show us is that this copper lever works systemically. It extends way beyond just basic skin repair. If we look at some recent data, in the lungs of mice, it significantly calmed down inflammation by lowering TNF-alpha and IL-6 levels. In rat models looking at nerve repair, it actually increased nerve growth factor and those myelin-producing Schwann cells. And there's even a cutting-edge 2025 study on colitis showing it helped restore intestinal barrier proteins. So we are really looking at a single copper courier facilitating deep, deep repair across completely different types of tissue. Which brings us right to our ultimate reality check. Biology is elegant and it's broad, but human proof and tissue delivery, those are still totally unsolved. This right here is the open frontier. See, there's this massive gap between what a molecule does elegantly in a petri dish or a lab mouse and how it actually behaves when you try to apply it to a living, breathing human being in the real world. The biology of GHK-Cu in a computer model or a lab is undeniably brilliant, but it crashes headfirst into the harsh physical realities and limitations of cosmetic delivery. Because the core physical problem is simply this. GHK-Cu is hydrophilic. That means it loves water, and frankly, it's highly unstable. And just by pure physics alone, the lipid barrier of human skin is specifically designed by evolution to repel exactly this kind of molecule. A 2015 ex vivo study using human skin proved this perfectly. It demonstrated that without aggressive physical assistance, peptide penetration was practically zero. It wasn't until researchers physically broke the skin barrier using microneedling that they managed to get around 134 nanomoles of the peptide across. Bottom line, if you don't physically bypass the skin, this fragile little molecule just stays locked outside. And because of this fundamental hurdle of physics, getting this fragile molecule completely intact into human tissue is still very much an open research problem today. In fact, there's an upcoming 2026 systematic review that points out how stable formulation and deep penetration are the exact methodological roadblocks that are limiting its real-world clinical translation right now. So, at a pure high-efficacy level, GHK-Cu strictly remains a research-use-only compound. The biological mechanism? Unbelievably clear. But reliable, large-scale delivery in actual humans? That's a puzzle we're still trying to solve. So, I want to leave you with this final, kind of provocative thought to chew on today. Think about everything we've just unpacked. If the most potent and elegant repair signal is already natively written directly into your own collagen, just waiting to be released by your biology, what part of the massive cosmetic industry is simply trying and, honestly, often failing due to the basic physics of the skin barrier to sell you back exactly what you already have? The gap between flashy marketing claims and harsh physics is huge. The true miracle here isn't found in a jar. It's fundamentally wired into our own biology. Thank you so much for joining me for this analysis. Stay curious, and I'll catch you in the next explainer.